So welcome everyone. Our guest speaker today is Dr. Blaise Frederic from McLean Hospital. Blaise received his bachelor's in physics from Yale University, and he got his PhD in biophysics from UC Berkeley. He's currently an associate professor at McLean and Harvard Medical School, and he's also the director of the Technical Instrumentation Core at the Brain Imaging Center at McLean. His research has been focusing on developing multimodal imaging acquisition and processing tools. He recently has made some great progress of doing simultaneous NIRS and fMRI together, and to better quantify the physiological parameters and try to denoise both data. So, welcome.
Hello. As I was mentioning before, I looked at the title of the talk and I couldn't even remember it, so I thought I'd summarize it with finding hemo, which is essentially what we're doing here. We're trying to figure out ways to use NIRS and other techniques to unlock the hemodynamic information that's hidden away in your BOLD data. Okay, so just a brief outline of what I hope to cover. Just a little background. You know this. BOLD is made up of lots of different signals. I'm going to show you how time delay processing can be used to separate them into signals that move and signals that don't, and that both parts, the moving signal and the non-moving signal, are interesting in their own right. And then I'm going to show you a couple applications using this. I've moved signal denoising to the end, measuring cerebrovascular reserve, characterizing cerebrovascular pathology, and then a little note about things you should think about when you're processing resting state data. Okay, so as we know, the BOLD signal is made up of a lot of different signals lying on top of each other. There's local hemodynamic response to neuronal activity, which is ideally what we're looking for most of the time. And then there's this sort of garbage bag called physiological noise, which is everything that doesn't come from the scanner itself. And that includes low-frequency oscillations, cardiac effects, respiratory effects, just not only the breathing waveform itself, but downstream effects having to do with depth of respiration, changes in blood oxygenation. And there's also motion. Then there's instrument noise. And unfortunately, a lot of these have significant overlap both in space and in frequency, because their cardiac signal, for example, is very spectrally different from these other signals, but it's aliased so heavily in fMRI that it's spread all over the place. So I'm going to talk about trying to separate the four signals that I mentioned here, the local hemodynamic response, and then also the low-frequency oscillations, cardiac effects, and some downstream respiratory effects. So as I said, we can't separate the signals in space. We can't separate them in time because they exist throughout the entire time course. We can't separate them in frequency because of the aliasing. So what's left is that we know that blood moves, and we're going to use that fact to do all kinds of cool things. So global hemodynamic effects, anything that's a property of the blood is going to have to move with the blood. And if we can figure out a way to figure out how the blood is moving, then we can basically split these signals up. So just a brief background on MR flow characterization. Most time-of-flight techniques, which are what a lot of people use, they can all be summed up as you want to change or measure the MR properties of blood at some particular location, and that's called that's tagging the blood. And then you want to detect correlated change in the image somewhere else at some other time. And by looking at the time delay between when the correlations happen and the strength of the correlation, you can make some inference about how much blood is flowing to where and when. So what do we use as a tag? People have done lots of things over the years. I mean, the first thing, and obviously you did it here first, is paramagnetic contrast agents. Those are great. They have high SNR, they have very unambiguous timing. You can get nice clean signals. The problem is it's invasive. You have to give somebody a shot. It's expensive, and some people don't react well to the shots. They get allergic reactions, and in some populations they can actually die from the results of the shot. So you'd like to avoid that if you can. More recently, what people have been doing is using an RF label for arteriospin labeling. So essentially, you tag the blood with an RF inversion as it goes through the carotids, and then you watch the downstream effects of either inverting or not inverting that blood on the signal in various parts of the head. And that's really helped a lot in a lot of different studies. Unfortunately, it has a relatively low SNR. I mean, you're talking about a 1% signal change typically. And that's if you get 100% labeling. And the tag has a limited lifetime, and that's going to be important in some of the stuff I'm going to show later. Basically, if you invert the magnetization in the blood in the carotid, it's only going to stay inverted for a certain amount of time before it relaxes back. And if it relaxes back before it gets where you're trying to measure it, then you're not going to be able to see anything. The other thing is both of these tagging schemes require a separate experiment. I mean, you can do hybrid bold ASL experiments, but they're generally a little complicated, and you have to take a lot of compromises in the bold acquisition, especially in terms of TR. So what I'm going to talk about today is using the blood oxygenation itself as the tag that you track around through the body. So as we know, oxygen is bound to hemoglobin in the blood, and the oxygen concentration tends to fluctuate due to all sorts of things, change in demand, change in flow, and change in depth of respiration. There's many factors that affect it. And these changes in oxygenation tend to propagate throughout the bloodstream. You can imagine that they sort of have to be in phase at the heart as coming out of the aorta, and then they're going to go all over the place through the various arteries into the capillaries, venules, come back, and then they're going to go through the lungs and all get in phase again in the aorta, since all the blood has to pass through there. So when you think about that, that's going to lead to internal timeline correlations between every voxel of a functional image, because there's going to be some signal that's in one place at one time and then it's in another place at another time, because the blood that was carrying this particular pattern of oxygenation gets to different places at different times. And we can exploit this in order to figure out what component in the bold signal is the moving component. And the nice thing about this is these correlations are in all bold data. You don't put them there, they're just there. As long as you have flowing blood, and if you're not doing ex vivo work, you're going to see these. So the question is, I mean, you can probably see that what I'm saying is true, but is it useful? And is it something that we can exploit, or is it is it just an interesting curiosity? I think you can guess what the answer is, because I'm standing up here talking about it. And the nice thing is that it's strong enough that you can even find it if you aren't looking, which is how we found it, because we weren't looking. So this is all based on a serendipitous result we got in 2009, 2010. And I'm going to present this in a way that makes it look like we're intelligent and knew what we were doing, but in fact this was a result of several cascading mistakes that led us into an interesting direction. We were looking at some old simultaneous NIRS fMRI data that we had lying around from 2006. And what we had this idea that we had done a parallel analysis of these data sets, where we'd looked at the NIRS and we'd looked at the fMRI, and we'd said, how are these different, how are these similar, how can we learn things about the propagation of photons? If we take into account the photon propagation through the head, can we better explain the NIRS signal in relation to the bold signal we get? And then we had this idea, since we're measuring the actual hemodynamic response to some tissue at the surface of the brain, in this case it was a finger tapping task, so we were looking at the motor cortex, then we could just do away with this whole assumed HRF thing. We could just measure the HRF directly in a chunk of tissue in the center of the region that we're interested in, and then we can take that and use that as a regressor to probe our data, and we can find minuscule correlations with our motor cortex all throughout the brain. Or or we can basically make this a much more sensitive detection by getting around having to assume an HRF function. That's not a terrible idea, but it's not a right idea either. It doesn't work very well. But what we found out accidentally was pretty cool. So here's the setup, sort of standard bilateral figure 8 coil for measuring the motor strip. So here's what happened when we did this. If you look at the bottom panel, this is standard GLM analysis with a square regressor, tapping, not tapping, tapping, not tapping, convolved with a canonical HRF. And you see you get pretty good activation in the motor cortex. But we can do better, but no, actually we can't. When we take our regressor that we measure with NIRS, which allegedly is exactly the response that we're getting, we kind of see the response in the motor cortex. That's the middle panel there. But we get all of this junk all over the place. There's signal everywhere. And in fact, even if we take the signal from the contralateral side and the voxel that we selected from the NIRS to not have any activation to the task, we're getting about as much activation, if not more, in the top frame that we are from the correct regressor. So this is telling us that this is not a great analysis technique. But then we thought, oh wait, no, it's still a good idea. We just did our analysis wrong. So again, we're saying, okay, we see widespread bilateral symmetric activations all across the brain. They're similar in the task and non-task regions. But we know that NIRS can only see about 2 to 3 centimeters away from the probe at most. And a lot of the activations, as you can see, were in the base of the brain. They were clearly, this was not something that we were seeing with NIRS and then correlating with BOLD. So, we think it'd be some artifact, but really, BOLD and NIRS really don't share any artifact mechanism. I mean, they're completely different instruments that are not electrically coupled in any way. But we figured out that what we probably did is we messed up the synchronization of the data. Because we have 2 different machines recording on 2 different time bases with 2 different clocks, and we synchronize them with a signal that we sometimes don't get. And so we figured, we slipped a TR somewhere, and we're not lined up. And we do know that the GLM analysis hates time shifts. If you're off by a TR, you get terrible activation, even in the presence of a real response. And that's why we have, hemodynamic response functions and slice time correction and time-derivative regressors, is to make sure that we don't get misaligned between our data and our regressors. So we said, okay, well, we came up with all of these arguments for why. Oh yes, of course we should be off by 1 TR in the positive direction because this, that, and the other thing. And we tried that. It's like, well, we got something that looked different, but it didn't look better. And then we said, oh yes, no, I meant -1. It had to be shifted in the other direction. We tried that, and that looked different, but not better. So then we said, okay, let's try every possibility. We're probably not off by more than 20 seconds. So let's just go. We'll just step through every delay between +1 and +20 and -20. But we still continued to see these activations all over the place, and our activation to the hemodynamic response to the actual tapping task didn't get any better. So we said, well, we're going to have to figure this out. So let's sort them and put them together into a movie so we can see if there's any place that looks best. And when we did that, we said, well, it actually does look best at different places at different times, and it always appears to look best in the blood vessels, and it appears to look best in the arteries first, and then in the gray matter, and then it looks best in the veins. So we were really, the thing that really got us was that our biggest activation was in the superior sagittal sinus, which is not actually in the brain. And then we looked at a phase contrast angiogram that we'd taken, and we said, well, look, these really are lining up with the vessels pretty well. Maybe this has something to do with blood. So could this be global hemodynamic fluctuations? Because we know that physiological fluctuations are huge in NIRS signals. In fact, they're the biggest thing you see if you don't filter them out. And in BOLD, they're very large also. In fact, the reason they're probably not larger in BOLD is the cardiac's aliased all over the place, so you don't recognize it. You just think it's sort of generic noise, but it's really, to a large extent, cardiac noise. We know that NIRS is sensitive to oxy and deoxyhemoglobin changes, and we know that BOLD is sensitive to oxy and deoxyhemoglobin changes and total hemoglobin concentration. We also know that the moving signal moves like blood does, and it seems to be strongest in the draining veins at the end of the experiment. So we think we may have accidentally found a tag for tracking blood around. So we tried to do this analytically in such a way that we could actually prove this, because this was based on a couple data sets. Actually originally came out of a failed experiment to try to localize a task in the frontal lobe. We found out we put our NIRS probe in the wrong place, and we weren't actually over the part of the brain we thought we were, but we were still getting really good activations, but not anywhere near the task regions. So this one we actually put a probe somewhere where we didn't think we would see any activation. We were actually going for we're trying to do just pure resting state. So we put this signal, we put the probe on the head. We had a short distance and a long distance probe, and we looked at various combinations of the NIRS signal and the BOLD signal. And we decided to give our analysis technique a name. We call it regressor interpolation of progressive time delays, which has the nice acronym of RIPTiDe. Where essentially you try every possible delay over a physiologically plausible range, and in this case it's -7.2 to +7.2 seconds. And this is based on the fact that radiotracer studies that were done on the staph knees with technetium show that blood, from the time that blood enters the carotids to the time it hits the jugular on the way out, is about up to no more than 9 seconds. So we figured full transit time is not going to be very long. So we found actually that when we did this consciously with no task and with just pure resting state, not doing anything, and a nice clean NIRS signal that shouldn't be contaminated by neuronal signals, we got really good maps that look an awful lot like blood moving around through the brain. And we were kind of happy with that. And the timings worked out in such a way that they agree with what you would expect, as I say, from the radiotracer literature. So we thought that's great. Let's learn more about this so we can turn this into a useful technique. So the first thing we were thinking was, okay, so we're alleging that this is a global physiological effect. Well, if it's global, then it's not just in the brain, right? Because one of the things we found with this, if you look carefully at these images, you'll see that there's a bright spot in the frontal lobe right under the probe. And the reason for that is that despite the fact we're using a 1 centimeter short distance probe trying to only get superficial physiology, we did manage to accidentally, there it is. You can see it up there in the frontal lobe. We actually managed to accidentally get a neuronal signal. And in fact, it correlates in a lot of subjects. We also get the contralateral activation across from the probe, which means that we're looking at some sort of brain tissue and connectivity within the brain. So we didn't want that. We want to get this global signal. So if it exists in the brain and it's global, then it should exist in the fingers and the toes and the earlobes and the tongue and all of that. It should be everywhere. So we did a brief NIRS-only experiment to try to track this down and see if this was in fact true. And it is. So if we put an NIRS probe on your fingertip, and we put one on your toe, we put one on your left foot and on your right foot, we see a bunch of very highly correlated signals with only slight differences between them. And the slight differences are in the time delay, primarily. So if you look at the signal over there on the right, that's that's 2 traces from the finger and the toe, and they're awfully close. I mean, these look very similar. The difference is that the toe is delayed about 2.72 seconds relative to the finger. Now, if we look at the toes from the 2 opposite feet, they're actually usually within about 0.2 seconds of each other. So physiologically, this is making sense. It takes about your feet are about as far from your heart. One foot's about as far from the heart as the other foot, and it's farther than your fingertip. So then we started to go crazy, and we started putting these probes everywhere. So we put one on the cheek, on the zygomatic arch. We put them on the fingertip. We put them on the earlobe, and we kept seeing this. The cheek lags the brain, and by brain I mean we had the frontal probe that we had before. Cheek lags the brain by about 0.32 seconds. Fingertip leads the brain by about 1.76 seconds. Any part of the body seems to be highly correlated with any other part of the body with a time delay. So if you go back to your bold data and you take a fingertip signal, you get the same thing that you get when you record off the brain, and you don't get the hot spots. You don't get the hot spots under the probe. So this is nice. The other nice thing about this is that it's very easy to measure on the fingertip. It's just a single probe. So this is a much easier measurement to make, and in fact we do this routinely on most of the bold studies that we do nowadays in our lab. Because it's not invasive, we just throw this. We're going to put a pleth pulse on them anyway. The reason we can't use a standard pleth pulse is that if the plethysmograph that you have in your scanner has a high pass filter in it to cut out anything above, say, the respiratory band. As a result, you get a nice clean heartbeat, which is what you want, but you throw away the low frequency signal that we want. So further characterizing the physiology, we wanted to look at the different spectral components because, as you can see, that as you know, NIRS and BOLD have components relating to the cardiac signal, respiratory signal, and low frequency oscillations. And we think that they act differently as they go through the body. Low frequency signals, as far as we can tell, seem to move with the blood. They move about as fast as blood does, and that makes sense because if you're looking at a change in oxygenation, oxygen is bound to hemoglobin, can only go where the blood goes. There are also low frequency oscillations in blood pressure, and we think that also corresponds to movement of blood because if you have a bolus of blood, where you have more blood that's sort of inflating the arteries a little bit, that's also going to move only as fast as the blood that's carrying it is. Cardiac signals are different. I mean, they're essentially a pressure wave, right? So you're sort of pounding on the system, and it's a wave propagating through the system. So if we wanted to design an experiment to do this, to separate the signals, we'd have to get past the cardiac aliasing problem. In order to do that, we did a multi-slab bold study. So we basically did 9 slices with a TR of half a second, and then we did it again, and we did it again, and we did it again. And we did these stacks throughout the brain. And this works because what we're looking at is the correlation between the fingertip signal and the brain signal. We're not looking at the so we're looking at correlation, the time delay and the correlation between the noise. We're not looking at the noise itself. So different runs should be equivalent as long as you assume the flow is going to be relatively stationary between time points, and that seems to be a good assumption from our data. I should point out now that this experiment was done before we had access to a multiband MR sequence. Now we can do 400 millisecond TR whole head acquisition quite easily, and we've actually replicated this with that. So when we look, what we've done here is we've taken the red-orange signal is a signal you're used to seeing. This is a low-frequency oscillation signal. It's moving at the speed of blood. And then the cardiac signal seems to cover, similar paths, but much faster. And that makes sense, because sound waves move a lot faster than the medium that carries them. I think the heartbeat travels about 10 meters per second through the body, and I'm pretty sure blood does not. So the other interesting thing about the different paths is that the pressure wave is going to go, it's not always going to follow blood vessels, because, really, it can, if it pushes a piece of tissue, it can actually move the adjacent vessel. It can actually jump from vessel to vessel. It's probably not going to be exactly shaped like the low frequency signals, but we think it's going to be similar because of it probably will be bigger in the vessels. So this is a rendering of the image you just saw in 3D, and here it's pretty clear that the low frequency signal is going through the vasculature. And this lines up very well with simultaneously acquired phase contrast images. And you see the cardiac signal appears to keep repeating. That's because the cardiac signal is pseudo-periodic rather than periodic. Your heartbeat does vary somewhat beat to beat, but it's pretty much the same. One heartbeat's pretty much like any other one. So over a course of 10 or 15 seconds, if you correlate it, it's going to peak every time. It peak at every heartbeat frequency, but slightly less every time as you get farther away from zero. So that's why if you look at the time scale there, you see that the response is biggest at time zero, but it's certainly not zero throughout this entire range. Okay, so what can we use this for? So basically we have this endogenous non-neuronal fluctuation in the hemodynamic parameters, and that's about 30% of the signal in highly perfused regions of gray matter, and that compares to about 1% of the signal for ASL. So if you're going to be using this to look at a flowing blood signal, then you already have about 30 times as much signal to work with as you do with ASL. The other thing, which is very important, is that since the tag is a waveform of chemical concentration rather than an inversion or something, essentially has an unlimited lifetime. I mean, where unlimited means the time it takes you to go from the heart throughout the body and back to the heart again. So it's got one cycle's worth of coherence, which is significantly longer than the 2 seconds that you get with ASL. And then the question is, well, so if we split this up, what's the signal and what's the noise? And that depends on what you want to look at. So if we consider the moving component to be the signal, I mean, in conventional task-related or resting state MRI, this moving blood signal is the noise. It's the physiological noise that you want to get rid of. But we're going to say, okay, let's just call it signal and see what happens. Well, we can learn all sorts of things about the blood circulation because the blood, goes to the brain. We can see where it goes. We can see when it gets there. We can generate high-resolution RCBV maps. We can compare RCBV in different conditions. I believe we can estimate flow velocities and directions, although at the moment we can't do that because it's actually, just because you see blood arrive here at one time and here at another, it doesn't mean that it went from here to here. So there's a bit of a question of, I think there are ways to untangle this, but we haven't yet done that. And it's probably not going to ever be unambiguous. It's probably going to have to be guided by images of the vasculature or something. The really cool thing about this is you get this for free. I mean, you're just doing your standard bold experiment that you would have done anyway. And you have all this blood flow information. In fact, every single data set you've ever taken has this blood flow information in it. The question is, can you unlock it and get it out? So the first experiment, just showing you an application, is calculating relative cerebrovascular reserve. This is basically how much blood circulation, how much can your circulation increase in response to a challenge. And this is something that can, your reserve can go down in various types of pathologies. And it's a fairly simple measure to get an estimate of how well your vascular responsiveness is working. In this particular case, we're using the NIRS as a tag, so we're measuring NIRS in the fingertip, and we're doing a breath hold challenge. Now, people have done this before, certainly, by correlating the block diagram of the breath hold with the data that you get. But that's not necessarily the best way to estimate it, because first of all, it only really gets to the peaks. It doesn't fit the baseline very well. This is what the actual waveform in a voxel looks like. It looks terribly noisy. That's fine. We're used to seeing MR traces that look like that. But if we compare what a delayed block regressor looks like in a given voxel, if we look at the fit that we get if we're using a HRF convolved block regressor with data, you see that the match, certainly is large, but it's not great. And if you look at if you use a delayed NIRS regressor in contrast, you see that it matches very well, even all the little peaks and valleys in the baseline and in the peak. You get a much better agreement, so you get a much higher detection power using the true NIRS regressor. The other nice thing.
From the fingertip?
Sorry, what?
The NIRS is from the fingertip.
I don't remember. I can check. I think it's the fingertip.
And the signal in the brain. Where in the brain was that? Small ROI?
Oh, that particular one? I don't know. I'd have to ask Yunji. It was a typical voxel. It was not a super good voxel.
But in an area where you...
Somewhere in the gray matter.
It's in an area where you usually see those effects, isn't it?
Yes. Yeah. But the cool thing about this is that it also works in the absence of the breath hold waveform, right? Because we've demonstrated that we can do this with just the noise. So the fact that we can fit the baseline down here means that we don't actually need to have the peaks. So you can do a measurement with the breath hold and without the breath hold and compare them, because the breath hold is actually the challenge. The breath hold is going to change your flow and everything during the course of the experiment. So you'd actually rather compare to a baseline where you're not doing anything. And so we can do that. We can measure basically the strength of our vascular responsiveness. Now this is the peak. This is essentially the peak correlation at any time. So we're taking out the time delay here. We're saying that if we go to the maximum, the time of maximum alignment between our NIRS regressor and the voxel, we'll put the value of the correlation here, right? We do that for the breath hold and the resting state, and we just divide them. And you get this very nice map of the ratio. This is essentially a ratio of CBV in the one condition to the other. And we're able to get it. We're able to get sort of a percentage change in the blood signal because we're able to divide by the resting state signal, which you don't really have access to when you just do a standard breath hold processing. Okay, so that's all well and good, but how far can we take this? Can we use this in really difficult cases where you're actually trying to diagnose severe pathology? One thing that we've come to realize is that healthy controls are boring. Everybody's blood maps look pretty much the same. You do 100 of these and you start to hate them because they all look exactly the same. The blood comes in, it goes out, it comes in, it goes out, and everything's done in about 6 seconds. But we don't really care about healthy controlled circulation anyway, right? Because we know that their blood's getting where it needs to because everything's fine. So let's do it in people who have severe pathologies and see if we see different results. So in this case, rather than use the endogenous fluctuation, we're going to use an imposed oxygenation tag, and in this case due to a gas challenge. This can result in much larger changes in the bold signal than than the endogenous fluctuations you're looking at. And as I point out here, it's a really good strategy if somebody presents you with a huge data set of pathological population collected with a paradigm that doesn't have any NIRS. So if you could happen to come up with a way of processing the data, that'd be great. So what they did for these people is, these are people who are presenting to the neurovascular surgery service at the Vanderbilt Medical Center in Nashville. And these are people who have severe cerebrovascular pathologies, major strokes, moya disease, various other diseases like that. And so what they do is they do a standard battery of MR tests in order to assess circulation. And one of the ones that they do is a Carbogen challenge, where they give people a gas mixture. They alternate between air and Carbogen, which is 5% CO2 and 95% oxygen. The reason they don't use 100% oxygen is actually your perfusion can go down in that situation because your vessels kind of squeeze down. This actually enhances oxygen delivery to pathological tissue, which is why they were able to get it by the IRB. Because the initial response to the IRB was, wait a minute, these people are already sick and you want to give them something that's going to reduce their circulation. That's probably not a good idea. So what they did is they alternate Carbogen and air in 180-second epochs. So this generates an oxygenation time course, an oxygenation tag in the time course, which is huge. It's like 20 or 30%. You see it by eye. And this lets them know in people who have very impaired circulation, this signal is so big that if blood gets anywhere, they will eventually see it, which is why they use this enormous challenge. So we can use our processing strategy to look at this data. Now, first of all, you have to be a little careful with your regressor because it's not going to look exactly like the Carbogen challenge. I mean, first of all, these are people who are very sick. Their breathing is a little erratic. The gas takes a while to get through the tubes, and because generally, as I say, these people are sick, they don't spend a huge amount of time setting them up. They sort of throw them in and they throw the mask on them. They don't tune it and measure the delays every time. So you may have significant delays in arrival time between one peak and the next. Differences in shape because they may not have happened to start breathing on the particular time when you change the gases over. What we do in that case is we start from our sort of canonical waveform, which is this low-pass filter, which is the on-off waveform, and we correlate that with the brain using the RIPTiDe analysis method. And we say, okay, so we get lots of places that correlate with some degree of throughout the brain. We measure these correlation times, and we say, okay, well, these are what the peaks are. So we realign them all. So we line up all the time courses, and we run a PCA to find the shape that explains most of the variance. So this is trying to get to what the basic shape of the waveform is, and this includes all of the effects of the tubing and the patient's breathing and various things like that. So this is a better estimate of what the time course looks like, and we just feed it back and we do it again, and we run through this until we get convergence. It converges in 3 times. In every patient so far, it's come to a convergence in 3 cycles. And once it's converged, we have all kinds of interesting measures of cerebrovascular dynamics. We have the peak correlation strength. We have the time delay to maximum correlation, which tells you basically when blood gets where. And we have the correlation coefficient over time, which we can present as a movie. So as I said, we tested this technique on a number of subjects at Vanderbilt University and the Vanderbilt University Surgery Service. So we actually see what happens in pathological subjects. And the first thing we find out is that they're really, really different from controls, which is cool. Well, I mean, it's not cool for them. It's cool for us. So if you look, this is 8 controls that they ran through this protocol at Vanderbilt, and you see that the reactivity, you see that the blood flow maps look the same as all of the other ones I've been showing you. The signal to noise is a little higher. If we take a histogram of the delay times of when blood arrives where, we get the same distribution that we always get. It's about 3-second half width. Blood comes in, blood goes out. We've changed over instead of using z statistics, we're using r-squared correlations now. This is just we've altered the way we do the processing. This is much faster, and it's actually somewhat easier to interpret than the z maps. So the first application we have is we had a subject with a severe stroke in the right middle cerebral artery. So if you look at her histogram of delay times, first thing you see is that the half width of the peak is 10 seconds. So right away you can tell that there's disturbed pathology throughout much of the brain, but you also see that this tail goes way, way out to the long delay times. What that means is there's parts of the brain that are not getting blood until 100 seconds after they come everywhere else. So if we look at the correlation map, so this is the peak squared correlation at any time. So we see that the perfusion in the area of the stroke here is certainly decreased. It's dropped below the threshold. And if we look at the time to peak correlation map, you can see that actually within that region, the correlation time's up around 100 or 120 seconds. But the interesting thing is that blood does eventually get there. There is no part of the brain that we found where the blood doesn't eventually get there, but it gets there very, very late. Now, if you were to look at this with ASL, the question is, does blood get there in 2 seconds? Answer is no. So you would just see this void in the ASL when they have simultaneously collected ASLs, which are not actually all that useful because they don't, all of the blood in this entire region is just blacked out. The area of the stroke looks very large, but in fact blood is getting there at some time, and that probably has something to do with how well they're going to recover. So if we present this as a movie, it's much clearer. You can see the entire brain fills in, and then the area of the stroke gets blood. After it's left the entire rest of the brain, it's then only then showing up in the area of the stroke and going out. And you can see it never really reaches much of a peak. So there's not a lot of blood is getting there, but it is getting there. And you notice that peak is somewhere around, I think it's around 80 seconds. Okay, so the other group of people that they study there is a lot of subjects with Moyamoya disease. Moyamoya disease is a progressive syndrome where you get occlusion of one or the other of the carotid arteries, and this can cause all kinds of severe problems, usually in one side. So it sort of degenerates over the course of a year or so. There is a treatment for it, this long word. It's called EDAS revascularization, where essentially they take an artery that's not doing anything terribly important in the brain, and they sort of grab the end of it and they stuff it down into the part of the brain that's important, that isn't getting blood. And that works surprisingly well for something that's, basically, as long as the part of the brain that you need to get to is within the stretching distance of the end of that artery, you can treat Moyamoya fairly effectively. So we have a subject here who was imaged before her surgery, 6 months after, and then 22 months post. It says 12 months there. That's an error. So if you look at her vascular arrival maps, you see her right side is really messed up. I mean, the entire hemisphere is not getting blood when it's supposed to. It's getting it way, way later, 80, 100 seconds later. And again, on the ASLs, this just shows up in parts of the brain where blood never gets to. So you wouldn't necessarily know the degree to which this tissue is actually going to be recoverable. Now, this is one of the reasons that they do the Carbogen challenge there. That on the Carbogen channel, in the Carbogen signal, you can see that there is you can definitely see that there's a waveform. The waveform propagates to those areas, so they are getting perfused. But they didn't really have an effective way of processing it until we started working with them. So you see, after 6 months, circulation is getting substantially better. The blood is arriving and clearing faster. And after 22 months, you would not yet say her brain is normal, but the gross pathology is much, much reduced. This is actually a little easier to see on a surface rendering. The effect is particularly pronounced on the surface of the brain. We were hoping, actually, this is just a recent development in this. We were hoping that by doing mapping this onto a free surfer brain, we could actually predict from the images what specifically what deficits they have, what might get better, and what kind of deficits remain. So this is in preparation at the moment. So conclusion for that, the Carbogen processing seems to be a very promising avenue, and we're really going to try to push that as far as we can. It's fairly well accepted by patients. It's not like a gasping for breath kind of thing like a CO2 challenge. And the last thing is, so I've shown you an imposed tag with a Carbogen challenge. I've shown you a measured endogenous tag with an NIRS. Can you do it with just BOLD alone? The answer is yes. We can select a region that's primarily perfused by, that's just a blood vessel. Basically, you can stick a voxel in the superior sagittal sinus, for example, and you use that and you run it through the refinement procedure that I showed you in the previous slide or in the previous set of slides, and you can regenerate low-frequency blood regressor. It does work, but it's a little unstable for long TR data because you've got cardiac data aliased all over the place. If you use multiband data, however, it works really, really well. So if you have like a 400 millisecond TR, you can just filter the cardiac out before you do this, and you can generate nice circulatory regressors. And this is just an example of a data set I pulled out of the NHU ADHD data set. I'm going to say this is a typical data set because it looks good, but actually our failure rate's about 25% with this data set. About 25% of the time the cardiac contamination is just too bad and we can't pull a regressor that refines. It just sort of, the refinement procedure wanders off somewhere. Okay, so finally, if we consider the moving component noise, we can use it to try to denoise our bold data. So what we want to do is we want to try to generate cardiac and LFO regressors from the NIRS data that are optimally delayed and then subtract them out of the bold data. Having access to the unaliased NIRS waveform helps a lot in this because we actually know what the cardiac waveform is supposed to look like. And we actually, if we know the time delay when the cardiac signal gets to a certain voxel, we can basically replicate the aliasing that happens during the bold sampling process. So we can actually alias the cardiac signal in every voxel to match what actually happened when you did the sampling procedure. So you can generate regressors that look like garbage, but they actually correlate quite well with the bold data. This is not a new idea. Doug did this in several years ago using the NIRS, the raw NIRS regressor as a confound. And that increases the task activation for task activation Z statistic, and I think this is a finger tapping task that we tried. The Z stat increased by 1. So the next thing we tried is, well, what happens if we just take the regressors and we try 40 of them? We throw 40 of them in with different time delays. Our Z values increased by 2. Yay. But what's actually best is using the voxel-specific regressor at every time point. So what you do is you go through the procedure and you measure the time delay at every time point, generate a regressor from the NIRS data at every time point that exactly matches when you think the blood gets there, and you then use a GLM to filter it out of your data. The other refinement we do is we do split the cardiac and the LFO signals before we go through this procedure because as I showed you, they propagate at different speeds. So if you use the same time delay for both, it's not going to work out for you. Your cardiac signal is actually going to end up adding noise. We also use separate regressors for the deoxy and total hemoglobin waveforms because they do actually seem to independently affect the bold signal. The low frequency oscillations are complicated. I mean, there's not only oxygenation changes, but there are also probably blood pressure changes. And it's hard to disentangle which is which. So when we do this, we can get a median of about 10.5% of the variance out of the data compared to 6.9% for retro-ICOR. So it seems to work somewhat better. Some interesting features of doing this. We thought, well, maybe the total hemoglobin or the deoxyhemoglobin was the most important, and the answer is no. They seem to be both about equally important. They both take about half the variance out. Splitting them by time, splitting the different bands actually dramatically increases the way how well this technique works. And we compared it to retro-ICOR or RVT, and we did get significant improvement in noise removal. Okay, so basically you can see blood flow, and you can take noise out of bold data sets, and that's really good. I did want to say one interesting thing that's come out of this work regarding connectivity analysis. So if we have this big global low frequency oscillations, in sort of the 0.01 to 0.15 hertz band, that's just in the same band that all of your resting state networks exist in. You probably imagine, you can imagine that these are going to be a major contaminant to your signals, and they're going to cause a lot of problems when you try to quantitate things. So we tried to look at this by taking a data set of 10 participants. We ran a group ICA to get their major ICs, and we also simultaneously recorded NIRS of these people. And we used a dual regression approach to try to correlate the time courses for every resting state network we found with the fingertip data. We found that the distribution of correlation coefficients was not what you would predict from random chance. So basically, this red Gaussian here is what you would predict from random correlation coefficient. What we did is we took the data set and we time shifted 10 minutes into the future. So same subject, same spectral characteristics of their low frequency oscillations, but just doesn't happen to be lined up with the data. And we find that there are certain networks that always end up correlating very strongly with the fingertip signal, including sensory motor signal, and this one over here. Things like the development network actually are down here in the region where they're probably not that strongly correlated. I mean, they certainly have an effect from the global signal. So the top regressor is this one, which we all see this when we do resting state analyses, and we know that it's a circulatory one because it's primarily in the superior sagittal sinus and in the transverse sinus. It's in blood vessels, so you immediately throw that one out. This one, no, I wouldn't throw that out. I mean, that's probably has something to do with brain function. This one, we always see and we say, oh yeah, that's motion because that's the top of the head and the head is bobbing up and down because of cardioballistic effects, blah, blah, blah. It's not motion. It's not brain function. It's probably mostly circulatory. This is basically this is the network of draining veins on the surface of the brain. And you have a couple of those. Then there's a really interesting one, which is the anti-correlated white matter network. I have no idea why that does that, but that shows up in every analysis we've done. The white matter is anti-correlated with the fingertip, and it's not a question of a time shift because it's not a periodic signal. It's a pseudo-periodic signal. If you just multiply it by -1, that's not equivalent to phase shifting by 180 degrees. We don't know exactly what that's about, but you're talking about more than 50% of the variance, well, up to 50% of the variance in some of these important networks comes from this global physiological signal. Fortunately, if you do the procedure that I described in the previous step to remove that, you find that the correlation coefficients go back down to where they're supposed to. They basically fall right on a nice Gaussian that's equivalent to noise. You still get your resting state networks. They're still there. They're still big. But you've removed this source of confound that's coupling the networks together, actually. The only ones that really are seriously affected in terms of losing a lot of voxels are the circulatory networks that I pointed out before. The real neuronal networks actually tend to get bigger because this confounding signal tends to make the correlation, tends to make tends to make melodic not find them as well because there's all of this in-band noise on top of them. So basically, yes, it affects the quantitation of your resting state networks. But resting state networks really are mostly neuronal. There's someone in my group who is trying so hard to prove that resting state networks are an entirely physiological artifact, but so far he hasn't done that. The last thing I wanted to say is if you wanted to do this NIRS technique, currently we use an Imagent spectrometer, which is about a $100,000 device, and it's sort of cumbersome and it ties up people in my group to do this on every scan. So can you do this with something cheaper? Well, ideally you do it with the fingertip pulse oximeter. But unfortunately, on the Siemens system, that is we're not allowed to work. We're not allowed to change the frequency band that works in. It's going to discard the low frequency oscillations, and there's nothing we can do about it. But pulse oximeters are cheap, and there's some ones available that you can hack now. So we've made a full bandwidth pulse pleth pulse out of a pulse oximeter development board that you can get for $28 from Mauser. Now, why they make pulse oximeter development board? I didn't think it was an active area of research for most people, but they make them, and they're cheap, and they give you the firmware, and then they let you flash new firmware onto it. So we can actually change this to preserve the low frequency signal, and so for about $70, you can make yourself a full bandwidth 14-bit pulse plethysmograph. And so we check this against the Imagent, and we get the same signals, and they're really fairly indistinguishable, and we've used this to process some MR data, and it's just as good. Making the device MR compatible has been a bit of a hassle, but I think we've gotten it now where it's not going to fly in and kill anybody, and it's not going to disrupt anybody. So basically, in summary, with time delay processing, you can separate the moving and the non-moving signals to measure hemodynamics and remove physiological noise. It works with exogenous and endogenous blood oxygen concentration changes. You can get your tag many ways. You can pull it out of the data with NIRS using cheap instruments. You can get it from the node and waveform. You can get it from the bowl data itself. And even if you get a terrible initial estimate, you can refine it and make it better. We could even start refining the NIRS signals because there's no reason to believe the NIRS is going to be exactly equivalent to how those signals are manifested in the bowl. It doesn't require any special hardware or pulse sequencers, other than, as I say, a cheap pleth pulse. Although going to a very low TR helps. If you can go below 400 or 500 milliseconds on the TR, then you can routinely filter out the cardiac signal, which makes everything much, much easier for this. So, I just wanted to thank my group. Yunji Tong, who's done an awful lot of these experiments, my graduate student, Lea Haka, and Kim Lindsay, who is also who helps get all of these experiments run, and all of my collaborators at various institutions. Thank you very much.
Is there any questions, please?
That was a fascinating talk. Extremely important work you're doing. I'm wondering, you didn't mention it, and I'm sure that we could cover a whole hour new lecture, but what about people who regress out the global signal? Is that something good for your data or not good? Because you're seeing these global hemodynamic effects. Are you taking them out if you take that global signal, whole brain?
Right. Well, see, what
should you do it or should you not do it?
I would say you should remove the global signal, but our way rather than if you average the entire. I mean, you can actually, one way to generate a regressor for this is to generate the global signal average. If you run that through the RIPTiDe procedure, you will get a flow map. Not a very good one, but you could refine it and make better ones. As I said, the GLM hates. It really hates time delays. If you regress out a phase-shifted copy of a signal that exists in a given voxel, you may actually make things worse because you're not fully going to remove it, and you may alter its shape or give it a phase shift, which means that in areas where the global signal regressor is very strong at that time shift, you'll make things better, and in areas where it's time shifted by a lot, it will probably make it worse. So.
I want to ask one question. So you're saying the long TR, bold data, you had some issues because of aliasing from cardiac. Right?
Yeah.
So, so just to this question of could you use old data that everyone has, old data to pick this out? So if you had a pulse waveform,
Yes, actually,
could you
Yunji.
de-alias right the cardiac and then do your analysis?
Yes, Yunji has done those cardiac maps that I showed you.
Yeah.
Yunji has a manuscript in preparation where he's doing it with the Siemens blood pulse.
Right. So there's hope
there's-
to analyze old data to get this. Right?
Yeah. The other thing is, now that there's all of these databases, if you have 1,000 free scans, you can throw away 250 of them, and you're still ahead of the game. But of course, you have to process those 250 first, and it still takes a while.
Okay, let's thank Blaise for his awesome talk.
I just want to say hello. Kuna Van Dyke. I work in Randy Buckner's group. I do a lot of this resting state fMRI work.